So our next presentation will be from the Atlas of Variant Effects Alliance team. David Adams is going to come up and say a few words and then show you a movie.
Okay, so I'm Dave Adams from the Wellcome Sanger Institute, and I'm talking on behalf of the executive of the AVE Alliance. So, of course, the reason why we're all here today is really illustrated in this slide, as you can see. So just as the cost of sequencing has decreased quite precipitously over the last few years, the number of these variants of uncertain significance have increased. And this really represents a major challenge in terms of the way that we diagnose, treat, and indeed counsel patients and their families. So what we're interested in as part of the AVE Alliance is generating these variant effect maps. We're interested in approaches such as the ones you've just been hearing about that are systematic, that are comprehensive, and that are accurate. And of course, this can include approaches such as SGE, base editors, and of course, VAMP-seq. So the Alliance was founded in 2020 at the height of the pandemic. It's grown very rapidly. We're now at over 300 members distributed in 20 different countries around the world. The Alliance has several agendas. Firstly, we organise this meeting, which is being held this year in Toronto. Secondly, there's the variant effects seminars, which I know many of you have attended. And following on from this, there are several other primary objectives that the Alliance has. Principally, this includes bringing together the data generators, the data curators, and also the consumers of that data. So this community that is here today. Going forward, what we really want to do is be the centre of the group of people or the group of labs that produces these comprehensive variant effect maps for disease genes and ultimately for all genes in the genome to really power forward the understanding of genetic variation at scale. So how do we do this? Well, we use the approaches you can see here by setting standards, by coordinating efforts, and by sharing tools and information. And our values and principles are shown here, including inclusivity, open access science, research integrity and excellence, and these 5 R's that you can see shown on the bottom of the slide. So how is the AVE Alliance actually organised? Well, there are 4 interconnected work streams. The first of these, and you've already heard a little bit about this already today, is ETS, Experimental Technologies and Standards, and this is led by Melina and Victoria. Essentially, this group is involved in, as the name suggests, setting standards and the methods associated with performing these kinds of assays at scale. The second group, which is the AMP group, are involved in the analysis, modelling and prediction of data that comes from these MAVE assays, and Debbie and Ben lead that group. We've heard a lot this morning from Claire about the importance of really understanding the data that you get from MAVE assays. And so the CVI group, led by Leah and Claire, are really tasked with that job of taking the information, facilitating clinical interpretation of that data. And then finally, I mentioned to you that this is an open access alliance, and release of data is very important. As it should be for this community. And the DCD is led by Alan and Maria. So there are a number of resources that sort of sit at the centre of AVE. These include, of course, the website, but also the MAVE set of resources. This includes MAVEDB and, of course, the MAVE registry, where ongoing projects that are performed by members of the community are collected. And this can really tell you who else is working on your gene or has an interest in performing experiments on your gene going forward. So this is the makeup of the current executive of the AVE Alliance, with representation from a range of different organisations. Clearly, one of the things that we need to do over the next year or 18 months or so is to improve and broaden the representation of this group going forward. And we're very, very ably supported by Lara and Alex as part of the project management team. So I mentioned to you previously that the AVE Alliance is organised into these work streams, which you can see here, now with the photos of the individuals who lead those work streams. But what I didn't mention was the ODIC group, who are led by Willow and Irene. So this is here because we feel that this cuts across all of the other working groups that I've mentioned to you already, and it's really an important part of this alliance, the way that we work and the way that we operate. So earlier in the year, there was a survey that was performed, which was really asking the question, what are the impediments for you to move forward in performing these kinds of MAVE assays at scale? And what's shown here are the results of that assay, of that particular analysis that was done. So some of these we can't really help you with, for example, the effects of COVID, but there were some messages that came out quite clearly from the survey. The first of these was that training is important, and one of the things we've been talking about as part of the executive is running a workshop or a training experience that could be, for example, run at Cold Spring Harbor, or maybe as part of Connecting Science at the Wellcome Sanger Institute to help train the next generation of MAVE scientists. We've discussed a lot about reagents, how we can make them more freely available and to the community at large. And of course, funding is also important. So we've also been talking about bringing together a funding council to bring all of the various funders around the table to discuss how we can fuel this alliance and indeed the generation of these maps that are so important. So how can you be involved? Well, firstly, go to the website and become a member. You're very welcome to join the group. Secondly, you can tune into the Variant Effects Seminar Series, follow us on Twitter, and you can also, I hope, attend this meeting next year, which, as has already been mentioned previously, will be held at the Wellcome Sanger Institute in Cambridge. So thank you very much, and I believe now we have a set of videos that are from each of the workstream chairs who are going to tell you about what they're doing and what the focus of each of those activities is.
Hello and welcome on behalf of the Clinical Variant Interpretation Group, co-chaired by Leah and myself, and constituted by Fritz, Brian, Rachel, Alex, Tina and Jonathan. So this group, our mission really is to develop approaches whereby we can integrate the variant effect maps, the MAVE data with other sources of information for clinical variant interpretation, how we can establish best practice for benchmarking the clinical utility of MAVEs and variant effect maps, and how then we can segue into exemplifying application of this MAVE data into diagnostic practice. So what are we working on? Well, we're embarking on projects around standardizing truth sets, very hard work, but very important. Thinking about how we prioritize which MAVEs are of greater clinical importance and should be embarked on sooner rather than later. We're looking at establishing guidelines for combining MAVE data with other data, using intermediate MAVE data, and combining data from multiple MAVE assays, be they concordant or discordant, and then working with the other work streams and stakeholders for thinking about how best to identify assays which reliably predict clinical effect, somatic variant interpretation, and quantitative conversion of MAVE data to best represent strength of evidence for clinical utility. And over the last couple of months, we've reached out to a broad group of people and invited some interested parties to join the CVI. So we welcome Jeffrey, Sali, Rossio, Sujatha, Elizabeth, Mandy and San Ming, who bring with them not just a wealth of different experience, but also geographic representation. And I look forward to updating you further on progress next year. Thank you.
Yeah, wonderful. It's such an honor to be here along with Vicky today, even though we cannot be there in person, to kind of give an overview of our experimental technology and standards, or short ETS workstream of the AVE consortium. So as you can see here is a slide summarizing the current acting members of our workstream. We have a wonderful mixture of different backgrounds and expertises with different model systems ranging from yeast and mammalian systems. We have different experimental technologies represented in the respective labs here. We also have more junior members as part of the work stream, highlighting here Daniel Tabbitt and Rachel Wilson, and input from Illumina through Gary Schrodt on emerging sequencing technologies. Vicky and I, we have the honor to co-chair currently the ETS work stream. And Vicky, if you would advance to the next slide. our mission really is of our ETS workstream to resolve issues relating to guidelines and best practices for MAVE data to interpret human genetic variation. And I'm handing over to Vicky now, who goes a little bit more in detail about what we are doing.
Great. Thank you so much, Melina. So we've been working on sort of several categories underneath that central mission statement. The first is really, and this was started by Fritz and Prashant in the first year of the ETS collaboration. The first is really to establish a controlled vocabulary for describing variant effect mapping methods. And the motivation behind this is that we want to be able to provide, concrete structured information that's available with any data and analysis that's uploaded to MAVEDB. So we're working very closely with Alan Rubin from DCD in order to do this. So this year we've developed an outline within ETS. This was led by Rachel Wilson, who's a postdoc in my group. And then basically was expanded by gathering input across the labs from ETS and their trainees as well. Melina and I then met with Alan Rubin to gather feedback on the controlled vocabulary that we had put together. And we are planning right now to have a finalized controlled methods vocabulary by the middle of this summer to be able to provide to DCD for upload for MAVEDB. The second sort of category of work that we've been doing is to develop strategies to guide others' experimental design and variant effect mapping. And the major accomplishment that we've had has been led by Melina and Daniel Tabbitt and Fritz Roth, in which they wrote a really fantastic review article that sort of covered variant effect mapping planning from soup to nuts. This was submitted to AR in April and was also reviewed and worked on by Prashant and I. And then lastly, we'd really like to move toward outlining how we can present the content of an information resource to those outside of the Atlas of Variant Effects in order to really use this as a platform for education as well as for information dissemination. So we did present a proposed structure for central resource to the executive committee earlier this year, and we continue to work with a postdoctoral committee from across ETS in order to establish what we call a DMS fairy tale, which we think is going to be a really cool educational resource for folks who are interested in actually implementing these methods in their own laboratory, and is born out of an idea that started in Benny's lab, where they actually have written out sort of a storybook almost in order to help people understand how to do variant effects mapping. And then lastly, we will have a call to action to solicit nominations for new members in the coming months. So thank you so much for having us today. We're sorry that we couldn't be there in person, but we know it'll be a great meeting. Thank you.
Thank you.
Hello, my name is Alan Rubin, and I'm one of the co-chairs of the Data Coordination and Dissemination, or DCD workstream. Maria is my co-chair, and we're joined by Carol, Melissa, Helen, Julia, Rashid, Shannon, Jack, Sumaya, Kevin, and Alex. This is a fantastic group of people with diverse backgrounds and experience, most with expertise in clinical genetics, informatics, and databases, or both. We're all working together to facilitate the distribution of MAVE data so that it can be more easily used for clinical and non-clinical applications. One of our major focuses is guiding the continued development of MAVEDB, the MAVE database. We're doing this by creating data standards for MAVEs that will be adopted by MAVEDB and hopefully many other projects. We also have representatives from different groups who want to use MAVE data, and we're making sure that the data sets our community generates can be distributed as widely as possible and therefore have the biggest impact. We're using our combined experience to help build the best data resources we can that will be the most useful to AVE members and beyond. The big projects we're working on right now are building on previous work to fully define minimum information standards for MAVEs, creating protocols to map MAVE datasets to the reference genome in a way that captures the genetic background used in the experiment, developing data models for representing individual MAVE variants so that they can be shared and carry all the necessary experimental context with them, defining data quality metrics for MAVEs that will inform data generators and data consumers alike, and making sure that MAVE data adheres to fair and care principles. If you'd like to follow what we're working on, please check out our GitHub space, And if you're interested in standards development and would like to consider joining the workstream, please reach out on AVE Slack or over email. Thank you.
My name is Debbie Marks. I'm from the Analysis, Modelling and Prediction workstream. And our mission is to promote the development of computational tools and standards that the whole community can use. So I'll tell you a bit about the who, the why, the what, and where we are up to. The team is comprised of this group here, and you can contact them individually or us all together, and Lara will tell you more about how. The two arrows are on the two chairs, Ben Lerner, my wonderful co-chair, and the chairs will rotate. It's not something we'll have for life. So do come join us, get involved. Three areas that we divide this up into is thinking about, and they're not completely orthogonal, of course, are benchmark data sets and what we can do with those and why it's important to have those, evaluation metrics and prediction experimental design. So let's talk first about the benchmarks. Why do we want benchmarks? Well, that means taking these wonderful experiments and annotating those and grouping them so that they can be used by the various communities for their goals. And it really does depend on what the phenotype is. So annotating of the assay type and phenotype is, and quality is really important. So, you might be measuring thermostability, kinetics, interactions, cellular function, organism phenotypes, all of which are important to different groups. So what have we done so far and what are we doing? Well, we are annotating these and creating these special sets, which we encourage the community again to be involved in doing. We import this to the MaveDB. You can go check out the paper, and obviously to the database group. We also work with the clinical variant interpretation, which when we assess those kinds of Maves that have had those goals, we will be releasing those as we've done them on our GitHub. Second area, prediction and evaluation metrics. What are the metrics? Well, why? Because, putting some ideas out there about how you might need different statistics for different data distributions. You might need a very different statistics if your data is bimodal to if it's unimodal and continuous rather than binary. And also important are the dynamic ranges and how to interpret those. And those all affect statistics. How do we deal with those? And the modeling goals, well, again, this is really dependent on what phenotype you want to do. So we will think about what the evaluation metrics are for being able to predict thermostability, biophysical things, or clinical consequence. And of course, we should remember standards. What have we done so far? We're writing a review paper on tools and metrics. We will engage with CAGI, which is an existing competition for variant effect prediction for all sorts of phenotypes. And we'll obviously import into other work streams. Lastly, not least, experimental design. Well, why do we need it? Well, we can't do everything. So being able to design ahead which are the most informative experiments and accelerate the field more is going to be critical. And the dynamic range, designing not so much the technology, but where is the dynamic range important for the prediction? And of course, designing, if we want library design and multiple mutations. So what have we done so far? We've started to make prioritizing genes, and this might be, with evidence supporting which genes might be easier to use existing methods to compute the effects for which assays, and we're really open to suggestions on this. We remember that this is for the world and our community in the world, and not for us, and we do ask you to come join us. Thank you.
Together, Irene and I co-lead the Outreach, Diversity, Equity, and Inclusion Committee. This is a team we've built up over the last year with the goal of increasing the diversity and inclusion within the AVE community. So this means both those who are taking part in the different committees and who are helping guide this organization, but also those who are doing these assays themselves. And by diversity, we're including geographic diversity, racial diversity, gender identities, and many other categories as such. We're also interested in how can we build partnerships and reach out beyond the alliance and scientific communities to those stakeholder communities who this work should impact. And then also starting to try to explore the ethical and equitable ways about by which we can go about MAVE design, what questions do we prioritize, how do we do these assays, and finally, how do we communicate these experiments. And so what we're really trying to work on right now is starting to explore how these high throughput data sets can start to bridge the gap and fill in gaps for populations that are underrepresented within clinical genetic databases, and so of course can't be called with strong statistical confidence, variance in those communities. We're also really interested in trying to start conversations about what the impacts of these assays are in these data sets, and how we can do better, because we should always be searching to do better. One of the ways we want to start these conversations and learning more is by bringing on speakers within the different platforms we have within the AVE community, like the seminar series and the symposium. Finally, we're looking for members. If this sort of work interests you, please reach out to us. So Irene, I, or Lara, and we'll be happy to bring you on board.